Hi, I'm Ed Kreinek with the Streams Lab at the University of Waterloo. Thank you for taking the time to view this presentation, and thank you to SFS for the opportunity to be a part of the 2021 virtual meeting. My email is listed at the end of the presentation. Please feel free to contact me with any questions or to share ideas. As landscape and climate are rapidly changing, understanding the geographic distribution of species and ecosystems and what influences this distribution is gaining importance. This is evident by the growing number of large-scale studies showing up in the literature. In part, the growth of biogeographic studies is due to the increasing capabilities of GIS and the availability of large-scale environmental and taxonomic data, but it is also due to the recognition of the importance of biogeography. Transboundary rivers offer a unique challenge to biogeographic studies and to land managers. Data for rivers that cross state or province boundaries, or especially international borders, can be difficult to obtain. Once obtained, there is the potential for mismatched data types and collection techniques. Yet the need to understand the influence of river flows and ecology does not stop at political boundaries. Basin management by one political entity influences or is influenced by land management across the border. The Red River is one such transboundary river basin. The Red River Basin crosses 3 state and an international border and is an important source of water for municipalities, industry, farming, and recreation, which all influence the biota and ecology of the system. Lake Winnipeg in Manitoba, for example, is an economic and culturally significant water body that is deteriorating with nutrient loading from agriculture runoff, municipality wastewater as the likely culprits. Transboundary cooperation and research is essential to help protect this water source. This presentation is a look at preliminary exploration of invertebrate data for the Red River Basin and is a transboundary collaboration between the Streams Lab at the University of Waterloo, Environment and Climate Change Canada, the Minnesota Pollution Control Agency, and the North Dakota Department of Environmental Quality. To our knowledge, this is the first transboundary biogeographic study of the Red River Basin river system. Our objective for this preliminary study and this presentation is to 1. Determine if there are differences in benthic insect communities among the Red River Basin's political jurisdictions and ecoregions, and also what taxa might be driving those differences. 2. To determine if there are differences in the benthic insect beta diversity among political and ecological regions, and 3. To determine if beta diversity variation is associated with distance among samples. The Red River Basin is approximately 117,000 square kilometers, with 89% of the watershed in South Dakota, North Dakota, and Minnesota, and approximately 11% in the province of Manitoba in Canada. The watershed runs from south to north, from South Dakota to Lake Winnipeg in Manitoba. 84% of the basin is used for agricultural purposes, both row crops and livestock production. For this study, we'll be focusing on the data collected in North Dakota, Minnesota, and Manitoba. The basin consists of very flat topography and meandering streams. There are 3 level 1 ecoregions located within the basin. Ecoregion 5, the northern forest, Ecoregion 8, the eastern temperate forest, and Ecoregion 9, the Great Plains. The majority of the basin is located in a prairie biome, an ecologically important ecosystem in which biogeography of rivers is relatively understudied. Of the 566 total samples in this study, 499 of them are located in the Great Plains, mostly in the Minnesota and North Dakota, and a small representation in Manitoba. The northern forests and eastern temperate forests are represented by 48 and 19 samples, respectively. Samples were collected from 2000 to 2018 by the North Dakota Department of Environmental Quality, the Minnesota Pollution Control Agency, and by Environment and Climate Change Canada and the Streams Lab. Multi-habitat sample collection was similar among agencies, with the main differences being a slight difference in mesh size and using a traveling kit versus 20 combined habitat sampling efforts. Data was reduced to insect taxa and adjusted to genera when possible, but left at the subfamily for Chironomidae and the family level for other difficult to ID taxa, such as Ceratopogonidae. All data was Hellinger transformed before analysis. When we consider ecological regions, the temperate forests and the northern forests have a higher richness than the Great Plains, which is impressive considering the temperate forests and the northern forests have about 70 sites total, and the Great Plains is a combination of 500 sites. In terms of political jurisdiction, Minnesota has the highest richness, followed by North Dakota and then Manitoba. Recall that Minnesota is comprised of all three ecoregions and the greatest number of sites, followed by North Dakota with about half the sites in only one ecoregion. Manitoba is only about 7% of the sites, and all but 3 of those sites are in the Great Plains ecoregion. If we use an NMDS, shown here, and ANISIM, there's no significant separation of ecoregions. However, even though the communities do not show separation, we want to know if there was specific taxa that are good indicators of these separate ecoregions. For this, we use an indicator species analysis. We set a minimum cutoff for specificity, fidelity, and the test statistic at 0.5, and only 3 taxa surpassed this cutoff. Simulidae, or black flies, and Leptophlebiidae, a mayfly, for the eastern temperate forests, and Corixidae, or water boatmen, for the Great Plains. All had moderate specificity and fidelity, and so none were considered great or strong indicators of any of the ecoregions. If we use an NMDS and ANISIM to look at the political jurisdiction, there is a significant but weak separation of regions. Pairwise ANISIM indicates that there is a significant difference between North Dakota, the green, and Minnesota, the brown. Because of the significant ANISIM, we used a simper to look at what taxa contributed to the community differences between Minnesota and North Dakota. Here I'm only showing cumulative contributions of 50% or less when ordered by individual taxonomic contribution. I think the main take-home point from these results is not necessarily what taxa is dominating the difference between the two, but more the fact that the difference among abundance is very slight for all these. In other words, there really isn't a large difference in these communities, even though it is significant. Having looked at the taxonomic composition of communities, I now want to transition to looking at the pattern of change in community similarity among sites, or the beta diversity. Beta diversity can be broken down into 2 components, turnover and nestedness. Turnover is the replacement of a taxa at one site by another taxa in a corresponding site. Nestedness accounts for the loss of a taxa between 2 sites without gaining another taxa to replace it. And together they represent the total beta diversity, or the change in community similarity among sites. In order to compensate for sample size among groups, we use the permutation of 10 sites for multi-site beta diversity. We found that total beta diversity is relatively high and similar among each ecoregions. Shown here is the density plot for the 10 site permutations with beta diversity on the x-axis and density on the y-axis. Note that beta diversity runs from 0 to 1, and this axis represents 0.7 to 1. In a similar fashion, we found that there was no difference in ecoregions for beta diversity turnover or nestedness. And also, we see that turnover makes up the majority of beta diversity compared to nestedness. When we switch to political jurisdictions, multi-site beta diversity is less in Manitoba than Minnesota, while North Dakota is not different from Manitoba or Minnesota. In a similar fashion for turnover, we find that Manitoba has lower turnover than Minnesota, while North Dakota is not different between the two of them. And we also find that nestedness, there is no difference between Minnesota, North Dakota, or Manitoba. And again, we see that turnover makes up the majority of total beta diversity. Finally, with distance among sites ranging from a few hundred meters to over 500 kilometers, we wanted to look at the association of beta diversity with site-to-site distance. To do this, we ran a Mantel correlation on pairwise beta diversity and straight line distances from site to site. We found that total beta diversity is weakly associated with between site distance and slightly increases as distance increases. There was no significant association of turnover with distance or nestedness with distance, although if you squint, it looks like nestedness drops slightly as distance increases, but Mantel did not find a difference. In summary, we found differences in insect richness among ecoregions and political jurisdictions. Insect communities are similar among ecoregions, but different between North Dakota and Minnesota. Beta diversity is mostly proportionate to turnover, and both total beta diversity and turnover is lower in Manitoba. And finally, beta diversity is only weakly associated with site-to-site distance. Our results help to demonstrate some of the difficulties in transboundary studies and leads us to our next steps in this study. What is it that is causing the differences among political jurisdictions? Are the differences ecological? Are they due to management differences? Or are they the result of sampling or data processing techniques? Once we can answer these questions, we can move on to others. There was limited association with distance and beta diversity. Well, what is it then that's actually driving the high turnover among sites? Does network connectivity play a role? How can we apply our new understanding to improve transboundary and management of the Red River Basin? Thank you for your time. Please email me with any questions or to share ideas. Thanks.